Our last speaker is Christopher Lewis. He's a postdoc at the University of Zurich. And the topic to finish this seminar, it's on the cortical resonance that has a selection role in coherent input. So Chris, whenever you want, off to you.
Great, thanks. Yeah, and thanks to the organizers and everybody for putting this together. So yeah, I'll talk about a resonance that occurs in cortex and how that resonance may affect transmission of signals through a cortical area. And most of this work was actually performed when I was at the Ernst Jungmann Institute in Frankfurt, Germany. But yeah, I'm now in the University of Zurich. And so rhythmic activity in the brain is ubiquitous. These are examples from Hans Berger of the first electrical recordings from the human brain. And there, one of the clear kind of, results from the data was that activity can be quite periodic. And indeed, here's an example trace where the subject had the eyes open up until when this arrow is presented, and then they close their eyes and you see a large amplification of a slow rhythm. So this is around 8 or 10 hertz. This is the cortical alpha rhythm. And subsequently, since then, oscillations have been found more or less all over the brain. And so they occur across quite a few temporal scales. So from very slow oscillations, even across longs of minutes. And so I think you can even think of circadian rhythms occurring across days or longer. And they also occur on very fast scales. So up to what's called like gamma band oscillations, which are in the range of kind of 30 to 80 hertz or so, and even faster than that for muscle-related oscillations, or for example, like hippocampal ripple oscillations, which occur kind of in the range of tens of milliseconds. And they also occur across all species and brain structures. So it seems like it's a quite ubiquitous phenomenon. They've been correlated with different cognitive or behavioral variables. For example, if a mouse in this case traverses a linear corridor, in the hippocampus, there is a strong theta rhythm, and place cells in CA1 will spike preferentially for given spatial locations. And those spikes will occur at preferential phases of the theta oscillation with respect to the mouse's position and trajectory in space. These same sequences can occur at faster time scales. So about 10 times faster, for example, in the ripple band when the mouse is at rest. And these sequences can reflect previous trajectories the mouse has gone, or also future potential trajectories. In other wake behaving animals, for example, cats or monkeys, also humans, if they're monitoring a visual scene, in this case cats, and an expected object is presented to them, you see strong alpha band synchronization in visual cortex of these animals, actually visual and parietal cortex. And you see gamma band synchronization as well, but it's quite reduced as compared to a novel stimulus. So some sense of violated expectation can give rise to larger synchronization in the fast frequency range and reduced synchronization in the low frequency range. So there's I think a large body of evidence like this that correlate synchronization or oscillations in different frequency ranges with different cognitive or behavioral effects, but there's still a lot of disagreement in the field about what the actual role of these oscillations might be. I think it even goes to the extent that many people believe that they're simply epiphenomenal in a sense, so they don't have any kind of causal role in the system. They simply occur when you have coupled excitatory inhibitory elements, and the oscillations per se don't actually facilitate any sort of information processing. So this is a question which is of interest to me because they are so pervasive. In some sense, Occam's razor might suggest they should play some role in the functioning of the system. And so one compelling proposal for the function of synchronization is actually in facilitating dynamically routing information in the brain, because depending on behavioral contingencies or goals, it can be the case that you don't want to simply propagate all information to subsequent layers in the cortical hierarchy, for example. So in vision, this is the case that you have a lot of potential sensory information, but higher order visual areas might not necessarily reflect all of that information, and information from the primary visual cortex might be selectively propagated according to its behavioral relevance. So you can imagine a monkey looking at some visual scene, and it might find two objects in that visual scene which are behaviorally relevant to that monkey. And these objects, that in this case the pear and the apple, will be represented topographically in area V1, but area V1 will then converge onto subsequent downstream visual areas that have larger receptive fields. So you end up with a situation in which the neurons in the downstream area have to, in some sense, decide which visual stimulus to represent. Or alternatively, they could kind of be in a bistable state where they reflect some part of each. And so one proposal that has been put forward is that when you have something like attention, that highlights object of interest, and that object is then selectively propagated to downstream populations. And there's a considerable evidence that this, at least that this does occur in monkeys, for example, from single unit recordings in area Emcee or V4, that not all information gets through, that there is some sort of informational bottleneck or some sort of flexible routing of information. And one possible implementation of that is synchronization. So if a visual object is on the screen, presented to an animal, so in this case on a computer screen, the monkey is freely viewing, this is data from Nick Brunet, an orange, then you get strong synchronization. This is a recording from area V1, and you see strong gamma band synchronization. So this is synchronization around 80 or 90 hertz. And this gamma band synchronization, we know from previous in vitro work and also modeling work, arises from the interplay between excitation and inhibition, because you have strong feedback inhibition from, for example, pyramidal cells onto PV interneurons, and then the PV interneurons can strongly inhibit the pyramidal cells, which give rise to kind of periodic excitatory periods. So this is kind of displayed here. So if you have some object represented in V1, you can have periods where excitation increases kind of the representation, you could say, of that object. And then that, once it reaches a certain level, activates the inhibitory network, which then suppresses spiking in the population. And then because inhibition has a time scale of some 10 milliseconds or tens of milliseconds, 20 milliseconds, then you get a rhythmic excitatory period, which then recurs in time. So if you have two visual stimuli, then you would have two different rhythms. And these rhythms would depend on physical characteristics of the stimuli, but also potential top-down signals, for example, behavioral context, and so on and so forth. And then, so these two visual signals then converge onto a higher order. So for example, area V4, and the proposal, which was put forward by Pascal Fries, the communication through coherence hypothesis, is that the population, which is synchronized to the downstream neurons, will be preferentially transmitted. So this is actually then some mechanistic role for oscillations in the brain. But so far it's been hard to causally test this, because you can find selective synchronization, and there's been quite a bit of evidence that V1 and V4 are selectively synchronized, for example, under attention. But you never know if the oscillations are there simply because they're, for example, arousal or excitation in the network, or if they actually play a role in the selective routing. And so we wanted to try to test that hypothesis. And so in order to do that, we expressed Channelrhodopsin in pyramidal cells of the cat visual cortex in area V1 and V4, a homologue of V4, which is area 21A. And we had expression really limited then to pyramidal cells. And this gave us control over the excitation in the local network in area V1 or in 21A. And so if we shine blue light on the cortex there in V1 or in 21A, in both areas essentially, we were able to induce strong gamma band synchronization. So this occurs around 40 to 80 hertz, depending on the strength of the excitation. But this gave us good experimental control over these oscillations. And if we look, this is the local field potential, and if we look at the spiking is very locked to the synchronous oscillation. So the pyramidal cells are basically becoming activated. This activates presumably feedback inhibition, which then gives rise to this gamma oscillation, which then subsequently locks the spiking of the neurons to the population rhythm. But we wanted to actually try to get causal evidence for flexible routing. And so in order to do that, we switched from using, this is a light which was static in time, so the light wasn't flickering, and the rhythm is purely intrinsically generated. And so to try to get a handle on causation in the system, we actually used white noise laser stimulation. So we flickered the laser with Gaussian distributed white noise with a mean intensity, which matched the constant illumination. And this temporally varying stimulation also gave rise to gamma band synchronization. But the advantage to this signal is because the white noise is completely uncorrelated in time, we can actually look at the effect of individual elements of input into the network on subsequent output of the network in terms of spikes in that network. And so to do this, thanks. To do this, we use the spike triggered average. And so what we do is we basically take every spike from units in the recorded population, and we align the elements of white noise to those spikes. And this allows us then to look to see if there were specific signatures in the white noise time course which gave rise preferentially to spiking. And in fact, we found that there was short bouts of gamma band input preferentially led to spiking in the network, suggesting that the resonance, the gamma band resonance of the cortical network actually gives rise to selective filtering of temporally varying input. So you can see that here. Also, instead of using a spike triggered average, we computed a statistical model between the white noise input and the action potentials of the population. And here you can see that really specifically in a relatively broad gamma band between, for example, 50 hertz and 120 hertz or so, we see the strongest effect of variance in the temporal input on spiking in the population. So it seems like the gamma band resonance is effectively selecting elements of the input which match its resonance. So to try to better understand this, we implemented some recurrent network models of excitatory inhibitory units, and these are simple point neuron but conductance-based Hodgkin-Huxley-like models. And if you give DC input to this network, it's well known that it generates strong gamma band synchronization. And so this has been studied by a number of groups, but we were specifically interested to see if the model would also reproduce the selective filtering effect we saw. And in that case, we actually then stimulate the excitatory units with white noise, and we see that there is selective transfer of the element of the white noise, which is coherent with the network resonance. And so it seems like this resonance does play some role also in the models in filtering it. And because we have the model, we can try to then understand the mechanisms of this. And so that's kind of what we do. We look at what is performing this. And so we isolate excitation from inhibition, and we only impose rhythmic inhibition onto a separate excitatory population, which is driven by white noise, which is independent. And here again, we find filtering. So it really suggests that the key of this dynamic or this filtering of the input is occurring through rhythmic inhibition. So it seems like rhythmic inhibition can basically preferentially transmit signals which match its preferred time scale. So that's the end of the story for here. And there's more I could talk about, but I think time's limited. And if you have questions, you can contact me at email or on Twitter. I'm on Twitter. And I think the people who worked on this with me, Jiangwang Ni and Thomas Wunderle and Patrick Indritza, all performed experiments with us. And Andrea Lazar helped with the modeling work, and Ilka Diester and Pascal Fries were involved in the supervision of the project. So
thank you very much, Chris. It was very interesting, in fact. I see there are no questions so far, so while they come, because we have like a few more minutes, I'm interested in the causal approach that you use. You use a white noise light versus the blue light. What are the characteristics for this causality?
No, I mean, the light was always blue. It's just that it's temporal white noise. So because the blue light is what activates the channelrhodopsin, right? And
yeah,
so then it's basically we either have it with no temporal structure, so like a DC or a step function, which then is providing some constant excitation into the network, and that generates a kind of intrinsic gamma oscillation. Or we flicker the light with Gaussian distributed white noise. So then we have a mean value which matches our constant illumination, but we have every sample. So in this case, it was around 1 kilohertz. We were drawing a new intensity for the laser. Yeah, so the laser was just flickering essentially.
because I guess all other causality methods that I know of, they are more invasive, whereas this one is quite elegant in a way. You perturb the signal on the cortical region, but you let it function as it is, right? So it's quite clean as a control.
Yeah, I think, I mean, so this has been used to understand single cells and things like this for like Panch-Klappen recordings. I mean, for example, I think Duncan kind of maybe alluded to this a little bit in his study, where you can patch a single cell and you can put white noise time sequences in. So people have used this to look at if cells have resonances, for example, or but then I think that applying this at the level of the intact system is very interesting. I think you could then study different circuits to see if they have different sort of transmission properties and stuff like that.
Okay. Since there are no more questions, I will have another. There are no more questions. I have another curiosity on this, more in the mechanistic part of the V1 and the V4 selection mechanism. Do you know, or do you get any gamma correspondence between both that can say there is a change between both regions in the object representation? Like, in other words, does the object from V1 to V4 have a somatotopic kind of transmission, or is it modified in the gamma that you see? I don't know if I was clear.
Yeah, I think so. I mean, there is evidence that synchronization is altered by, for example, like for example, attention. So if you have two visual stimuli, I think the most standard, there are two experiments I can think of off the top of my head. One from Conrado Bosman, where he had two visual gratings, which were in different parts of the visual field. And so in V1, they were activating different patches of cortex, and those converged onto some V4 population, which was then measured. So the V4 population represented both spatial locations, but V1 differentially represented those different locations. And under a single case, so stimulation with a single grating, that grating would be represented in V1 and then also presumably represented in V4. And you could see this through synchronization between the two recording sites.
Okay.
But if you had two stimuli in V1, the monkey, based on how its attention was oriented towards one grating or the other grating, which was done through a visual cue, would basically selectively synchronize with the attended object, and there was no synchronization with the non-attended. There's even a very elegant study of this also from Iris Grota from Andreas Kreiter's group. And there they also used white noise stimulation in the visual, but in vision. And so they basically had two patches on the screen, and the patches were flickering with different white noise time series. And so then you could see how attention selectively transmitted the flicker from the attended stimulus versus the non-attended stimulus.
Yeah. Okay. Cool. Well, we're gonna get kicked by the next session. So thank you, everyone. Thank you, the speakers, for these nice and interesting topics.
Thanks.
Have a good afternoon, everyone.